Thanks. Kia ora everyone. I'm Hamish Dunham from Wellington, New Zealand. So I'm here to tell you today about my PhD research where I developed a multiplex aptamer biosensor for detecting multiple amino acids. So in recent times, amino acids have been gaining interest in looking at plasma-free amino acid concentrations as markers for disease. So with this as an example, this here is a normalized sort of baseline for a healthy individual and what their amino acid concentrations will be. You can see in stage 1 lung cancer, there's an increased circulating amounts of pretty much all amino acids, and then as it develops to stage 4, it collapses in, particularly for asparagine, glycine, histidine, and tryptophan. So the circulating amino acid profile can act as a sort of thumbprint for particular physiological states. So that's led to this idea of creating an APTA index, where you pretty much just get a whole bunch of samples from healthy and diseased people and create a big index of all of the amino acid profiles. So currently the best approach for determining amino acid profiles in biological fluids is using GCMS, LCMS methods, which are technically complex, require expensive devices, and also derivatization of amino acids and other challenges. So one possible solution is a electrochemical aptamer-based biosensor. I'm not going to go into the details of how they work, but I've been developing them. You can see here with this result using a phenylalanine aptamer, when you add 100 micromole of phenylalanine, you get an increase in electrochemical signal. So the objectives of my PhD were to do CLEX on 4 amino acid targets and then characterize these aptamers and then develop a multiplex electrochemical aptamer biosensor using them. So the 4 amino acid targets that we chose were asparagine, glutamic acid, proline, and arginine. These were actually chosen because of their importance in bovine fertility. My primary supervisor is a reproductive biologist, not an aptamer researcher. So I used a conventional affinity matrix CLEX, where I conjugated the ligand to a bead. So here's just a diagram showing a kind of loosely bound aptamer sequence, a tightly bound one, and then one that has affinity for the mobilization matrix as well. So here's the CLEX scheme that I used. I did 10 rounds, a couple of positive enrichments, and then after that, some pretty intensive counter selection. The astute among you will notice that I'm using biological fluids. These biological fluids are likely to contain the free amino acid targets that I want to enrich my library against. This was highlighted early on when I was doing the CLEX, but we decided to just do it anyway and see what happens. And so this is what happens. We can see that there's not a lot of enrichment from the glutamic acid CLEX. There's pretty much none at all. Same for proline, same for arginine, but interestingly, the asparagine population did show a reasonable amount of enrichment. So I did NGS sequencing on my enriched libraries and then just ran it through the fast aptamer pipeline. I also incubated the library with a control bead that didn't have a target attached to it and sequenced that. And then anything in the control library that was above 10 reads per million, I subtracted that even if it was present in the target library. So that way I just was selecting sequences that found the target. So, in reality, what I think happened is I deselected for aptamers that actually have affinity for the free conformation of the target, and instead enriched aptamers for that had affinity for the bound version. So after you select your candidates, because I just selected them based on frequency, you need to characterize. So the characterization methods that we had in place in our lab group at the time, I didn't deem satisfactory, so I went and developed circular dichromosome ones. So I used the cocaine aptamer as a positive control because I'd been reading a lot of papers from Philip Johnson's lab. So I should use the pointer for the people online. So we can see here for MN4, which has a stable structure in the absence of the ligand. We have this big peak here, and when you add the ligand, it stabilizes a little bit. And then for Mn19, which has a destabilized structure in the absence of the ligand, this peak is not as high, but when you add the ligand, the peak amplitude comes to match what we see for Mn4. And you can see here that the structures match. So, I was pretty happy that yes, CD spec actually works. And then I also developed ITC protocols for our lab, because for small molecule aptamer characterization, it's just what's the gold standard in my opinion. So it's really useful because you can determine a near complete thermodynamic profile, and that by looking at the characteristics of the thermodynamics, you can determine if there's a large scale structure switch occurring, which is beneficial for biosensing purposes. So from all of this, I identified a novel L-asparagine binding aptamer. Because it was isolated using a conventional Celix approach, not a capture Celix approach, the CD spec shift doesn't show much of a change in structure. And then the ITC also doesn't show a lot of heat associated with the binding interaction. So this was 100 micromole aptamer with 2 millimole ligand in a low volume ITC machine. So because I'd already hydrated it to 100 micromole and I had some spare, I just increased the ligand concentration to 10 millimolar and got this very steep binding curve, but a binding curve. So I know that it works. It's just not a very performant aptamer, but that's because I deselected for its affinity for free L-asparagine. So yeah, using a low volume ITC required optimization. So on the right is a figure from Phil Johnson's lab, and I just replicated the experiment that accounted for the difference in sample cell volumes. So because I only managed to isolate a couple of aptamers for asparagine, it wasn't sufficient to make a multiplexed electrochemical biosensor. So I turned to the literature. So I found the ARG1228, which binds arginine, TERP3A1, which binds tryptophan, and then Phe1 and Phe3, which bind phenylalanine. I hadn't read much at the time, but I've come to learn that the ARG1228 can be truncated down to a 28 mer, and that's the L-arginine aptamer, which is probably the most published amino acid aptamer, but I'm going to die on my cross that it's not an amino acid target. So yeah. So I also put these aptamers from the literature through the characterization pipeline. I did test a broader range of amino acid aptamers that were in the literature, but there's only so much time and funding. So the ARG1228 aptamer showed binding in ITC, and it is salt dependent because it's driven partly by electrostatics, which has been previously demonstrated in the literature. I didn't really see much of a shift in the CD spectra, or I didn't see any at all, except for a small one here to tryptophan, some off-target interaction. For the tryptophan aptamer, again, the ITC, worked to give a really good binding curve. And then the CD spec shows a really good shift in response to the ligand. And from the CD spectra, it looks like it adopts the G quadruplex, which has been reported by Yang et al. In the initial paper. So the Phe1 aptamer, I'm just going to stand here and really advocate for this one. It works insanely well. It's always great when they do what it says on the box. The Stojanovic lab has just been putting out some good aptamers for the last 10 plus years. So yeah, an ITC, because it has a very noticeable structure switch, there's a lot of energy associated with the binding interaction, which makes it very easy to see an ITC. And on CD spec, we can see there's a big CD shift because of the structure change. The Phe3 aptamer, on the other hand, while it does show a shift, it's not as intense as the Phe1 aptamer, but the ITC is also really good. So now that I had this bunch of aptamers that I could actually work with, it was time to move on to making the electrochemical biosensor. So this was the setup that I was using. Just a microx connector with these, this is a single plex electrode. And this was connected to a Palm Sense 4, which was connected to a laptop. So just a, benchtop prototype. And then this was the multiplex electrode that I used. So the aptamer gets dotted onto each of the 3 dots there as the working electrodes. So 1 aptamer per working electrode. So initial results using the single plex electrodes, I'd done some optimization and found that 400 micromole aptamer at 800 hertz with 0.5 amplitude gave a good signal. The ARG 1228 aptamer gives a small amount of signal at 10 millimolar concentrations. The reported KD for this aptamer is 2.4 millimolar, so it makes sense that we're not seeing anything at around 1 millimolar, and then we are at 10. The tryptophan aptamer gives a marginal response, especially at high concentrations of tryptophan, which is kind of counter to the reported KD. The phenylalanine aptamer, again, as I said, it's just very performant. Straight out the gate, it just gave a 30 to 40% change in the electrochemical signal. And then the Phe3 aptamer, which looked really performant based on CD spec and ITC, wasn't as performant on the sensors. So what's interesting to me about this is that you can use ITC and CD spec and they can tell you, yes, this should be good for sensing, but maybe the geometry of the structure switching isn't actually suitable or the most optimal for sensing applications. So once I determined all of how they each work on the sensor, I then moved to the multiplex electrode system. We were having some trouble with having the signal be a bit noisier when we moved to the multiplex electrode system. One solution was to decrease the amplitude. However, when we did that, suddenly my arginine and tryptophan channels stopped working. As you can see here, they're just flatlining, but the phenylalanine channel retained its function. So from this data, I plotted a binding curve and I determined a KD when it's attached to the sensor is about 60 micromolar. So the reported KD is about 10 micromolar, and that's what I found with ITC as well. So, it's a pretty close approximation between the two systems. And I looked at some off-target effects. Again, only the phenylalanine channel was working. So it has some non-specific binding for tryptophan and tyrosine, the two really structurally similar amino acids. So due to time constraints, I didn't really get to look much at the asparagine aptamers that I'd identified. So a future direction is to look at truncations and other variants on that and to further characterize it. Truthfully, the current state of amino acid aptamers is quite limited, especially single-stranded DNA ones. There was a lot of interest in this research back in the 90s because of the implications in molecular evolution, but seems to have waned. Further optimization of the EAB sensor. I spent a lot of hours just manually dropping a droplet of solution onto the sensor and then taking it off again. I'd be really keen to incorporate microfluidics and save a lot of time and stress. Also, after I'd sort of begun this whole part of the project, I came across this paper by Philippe and Kevin Blacksco, which is a really good paper for how to maximize the signal of this thing and how to optimize it really well. I had done some optimization, but the way they outline how to do it is just really good. So I'd like to be able to go back and do that. I played a bit more with the multiplex sensor. We dropped the amplitude down to 0.2 because we're having issues, but I've since tried it after that. I don't know what we changed, but we fixed it. So it works at 0.5. So I'd be keen to try it again to see if the tryptophan and arginine channels become activated, but again, time and money constraints. So yeah, I just want to thank all of you, and I would like to thank my primary supervisor, Janet Pittman, in the Pittman Lab Group, my secondary supervisor, Jennifer Soundy, Yasmin Liu from who I worked with and I'm now working with at ESR. She's our electrochemistry expert. I really could not have achieved this without her, so massive shout out to her. And my good friend and colleague, William Odie, and Oren Robb and Zach Murray from the Pittman Lab Group. So thank you, and questions.